discussion, and it also sets us up for introducing our speaker, Daniel Zommick from Valent Biosciences, where he is responsible for commercial support and product training on the BT portfolio. Valent Biosciences is one of the largest BT manufacturers in the world, with products registered in more than 65 countries. He's going to be joined by Dr. Christopher Moses, and I also see Katie there as well from Valent Biosciences. Christopher is actually the biorational expert for Sumitomo Chemical, which is the parent company to Valent Biosciences in Asia. So they will be helping to answer any technical questions that come through. So please keep them coming through, because they are there and ready to answer your questions. So without further ado, I'd really like to introduce Daniel. Bring a wealth of experience. It's great to have you today, and we're really interested to have this presentation on the management of fall armyworm with sprayable BTs. So welcome, Daniel. I'm just going to turn my video off, and I'm going to ask you if you could load your presentation.
All right, thank you so much. I think you have to stop sharing your screen.
Yep. Perfect.
Here we go. All right, you can see it?
Yep, just need to maximize it.
All right, there we go. Well, excellent. You know, I want to start by thanking Allison and the Grow Asia organization just for this great opportunity to introduce you all, or maybe remind you about some interesting things about Bacillus thuringiensis. Today, more than ever, commercial BTs are a really important tool, not only for managing fall armyworm, but also for other caterpillar pests. As society moves towards a desire for using safer, lower risk pesticides, having products like BTs on the market is really critically important for our future. And so I'd like to start maybe stepping back a little bit with just a reminder about what a biocontrol is. We are very much in a very distinct group here in biocontrol, in which biopesticides are defined as maybe macroorganisms like beneficial insects, microbes like what we'll talk about today, natural substances like plant extracts, and semiochemicals like pheromones. And actually, Roma gave an excellent introduction to biocontrols. If you go to this link here, the Fall Armyworm Action website in the video section, there's a really good introduction to all of these different biocontrol tools. But today we're going to focus really just on the microorganisms. And in general, there are 3 general classes of microbial bioprotectants. The first are fungi. These are microbes like Metarhizium or Bavaria bassiana, bacterium like BTs, or viruses. Now, this image here on the left shows the different ways in which these different classes operate. So, for example, BTs and viruses exclusively work through ingestion, while fungi can bind to the fungi will attach to the outside of the insect and actually invade them from the outside in. And that's really an important aspect about microbial biocontrols. It's that they operate very differently from chemical controls. So, for example, microbial biocontrols are microorganisms that may or may not have secondary compounds, while chemical controls are very defined active ingredients. Microbial biocontrols are often have a complex mode of action. So, for example, BTs, we'll talk about it as a stomach poison today, while chemical controls have a very singular mode of action. For example, having a very discrete target site. Microbial biocontrols can be really difficult to define what the active substance is, as it's the concoction of lots of different things, while chemical controls always have a very clearly defined active ingredient, active substance. Microbial biocontrols also oftentimes are slow to have its effect, slow to kill the insect, while chemical controls can be very quick, having very good knockdown activity. And so what I think it's important to mention here, and what I want to draw you to, is the fact that when we're talking about microbial biocontrols and about BTs specifically, I want you to think about them not in the same vein as you would traditional chemical controls, but to consider that it is a living organism, and it operates within the insect in a different way. And why it's important to keep that in mind is because that also then helps inform us about how to use the products most effectively, and to use that natural mode of action to our benefit. So Bacillus thuringiensis is very much a targeted bacterial disease of caterpillars. If you look on the left-hand side here, this is a single cell of BT, which contains a viable spore that's kind of like synonymous with a seed from a plant. And there's that beautiful crystal shape, that bipyramidal shape, which are the cry proteins. So these are crystal, they're named after the shape, the crystal proteins, which are actually insecticidal proteins that will naturally crystallize within the cell. They're actually produced at such high quantity and at such purity during the fermentation process that it'll just naturally produce these crystals. The primary mode of action is to form pores, which will then inhibit feeding and eventually kill the insect. BTs also contain a viable spore, which, as I mentioned, is synonymous with a seed from a plant. These spores will germinate in the insect gut, not on the plant or in the bag or the bottle. It does contribute to mortality, but as I'll show you later today, the spores themselves are actually not toxic to the caterpillars. There are other soluble synergists, some of them directly toxic, some of them not, to caterpillars. For example, VIP3A, the vegetative insecticidal proteins, has a similar mode of action to cry proteins, but a very unique target site. It's also soluble and doesn't form those beautiful crystals. There's also secondary compounds that can synergize cry proteins, like PD100, another name for zwittermycin, and other enzymes and byproducts that are produced by the bacteria. So here is just a perfect example of how complex these how complex this product can be. So when we consider what actually goes into the sprayable BT, so what goes into the formulated product, it's all of these different components put together. And just to sort of to drive that home, this is an SEM image of what the components of the commercial BT look like. And if you'll see, there's sort of a rounded there's these rounded objects. Those are the spores, and if you look closely, you can find those beautiful bipyramidal crystals just spotted throughout. And so, in a commercial BT product, we have a very concentrated. We have a very concentrated. A high concentration of these spores and crystals, which we're spraying out onto the plant. Obviously, you can't see the soluble components here, but this is really the bulk of the products when you look under a microscope. Now, BTs are really commonly used throughout Asia, throughout the world. And they may, in fact, be more common than you might think for those folks that haven't used them before. For example, BTs are commonly used in cabbage or leafy greens as an important resistance management. So that is, as insects become intolerant to other insecticides, commercial BTs can still effectively control them. In the case of pineapples, for example, commercial BTs are used as they do not contribute to chemical residues on the fruit, ensuring that there are no issues with exporting those produce. In the case of oil palms, there's a really interesting and unique use of BTs to control the bunch moth, caterpillar that will eat the actual fruit themselves. And in this case, there's an important pollinating weevil. The targeted activity of these commercial BTs can actually very efficiently control those caterpillar pests, but have no effect on the pollinator weevil because they have such a selective mode of action. And finally, mosquito control. The commercial the Israeliensis strains are actually are incredibly are safe enough to be used actually in populated areas, in drinking water and things like that, to control mosquitoes, the mosquito larvae that can survive in that standing water. So in this way, commercial BTs may be more common than you might have thought. And actually, they've been around for a very long time. So commercial products have been used for more than 50 years. Last year, we celebrated, and today we're still celebrating, the 50th anniversary of Dipel. That's the flagship BT Kerstaki strain sold by Valent Biosciences. It was first registered back in the 1970s. And so today we're celebrating 50 years. And if you think back at what chemistries were around 50 years ago, few, if any of them, have really survived and are in common usage today. And that's because there's been a shift in what we want out of our pesticides. And that shift has really maintained commercial BTs like Dipel into today. And also today we're really distributing more BTs than ever in our history, which just goes to show that that the demand for these lower risk targeted insecticides is really high, and that farmers are seeing the efficacy. Now, with respect to fall armyworm, commercial BTs have been used as a tool against fall armyworm in the Americas really for decades. Excuse me, the fall armyworm is native to Central and South America. And so actually in the early applications of commercial BTs in the United States, early applications of Dipel, were oftentimes targeted to corn, where they could be used to control the European corn borer, but also other incidental pests like the fall armyworm. In Brazil, back in the 2013-2014 season, more than 4 million hectares of corn, cotton, and soybean were sprayed with commercial BTs to combat the invasion of the Helicoverpa armigera. And so, these products are really well known within the Americas to control caterpillar pests in corn, cotton, and soybeans. And so, I think by the end of this session today and the next one in June, I hope you have a pretty good idea about how they'll be able to be used effectively throughout Asia. Now, there are a number of common reasons why folks will use commercial BTs. The first is that they are effective. We see consistent efficacy, and if used at the appropriate time and at the appropriate rate, they can be similar to chemical controls. They have a low environmental impact as they're safer beneficials, and also they are a natural part of the environment. BTs are really all around us. They have an excellent worker safety profile, which means it's low risk for applicators. And they're also important for chemical residue management. So there's no maximum residue levels anywhere in the world. And finally, and maybe most importantly in the case of fall armyworm, they're an important tool for resistance management, as commercial BTs have shown no cross resistance with any other insecticide class, period, full stop. It's really remarkable how these products are so different. So you may recognize some of these attributes as things that are important to you, but what it really comes down to is that we have this a very effective but natural substance that that represents a low risk for applicators and has a lot of implications for how produce can be sold and used. Now, Bacillus thuringiensis in agriculture actually gets very specific, so down to the strain level. So what you'll see when you look at a BT label is you'll see that all the products are labeled as Bacillus thuringiensis, but there are two subspecies you'll often see. These are either the Kerstaki or the Azawi type strains. Now, within these subspecies, there are individual strains and then production strains isolated from those. So when you buy a bag of Dipel or a bottle of other BT products, it is only a singular microorganism that's in that bottle. So we produce at 100,000 liter scale a single microorganism, kind of like you would ferment beer. You're growing that singular yeast. Now, each production strain has a unique mix of insecticidal components that I'll go into more detail with momentarily. And because those components can be so different between strains, each one is actually registered as an individual active substance. So the strain ABTS 351 is considered a different active substance from the 1857 strain. And that's because those attributes are so different. And that's another reason why we really need to understand the complexities of microorganisms and consider them differently than chemicals, in that each one has its own unique properties, its own unique value. Now, there are different subspecies used for other uses. So, for example, we've talked about the Kerstaki and Azawai type strains that are used for caterpillar control. While there's a subspecies Tenebrionis, the BTT, that's used for controlling beetle larvae. The BT Israeliensis strain is used for mosquito and blackfly control. However, this is very important. Only the subspecies Kerstaki and Azawai can be used to control the fall armyworm. Oftentimes, in my experience, there can be a misconception about which of these types of strains can be used for what types of insects. But if you just keep in mind that only Kerstaki and Azawai type strains are effective against fall armyworm, you may find the Israeliensis out in the market, but it is not at all for this particular use. Now, these commercial BTs are actually effective against almost all agronomically important pests. I have here listed out just some of the pests that are on the commercial labels for the Sumitomo chemical products that are available throughout the region. For example, diamondback moth, nettle caterpillar, or bunch moth on the oil palm, as well as cotton bollworm, beet armyworm, and of course, fall armyworm. Now, there are a few insects that are not controlled by commercial BTs, and those are pests that either immediately burrow into a plant, or those that are actually hatched within a plant. And that's because commercial BTs are not systemic. You saw in that image at the beginning, these are just actual crystals and spores, a physical thing. Not necessarily a soluble metabolite. And so for that reason, they cannot move within the plant and they cannot move across the plant. So the insect will have to eat the foliage on the outside to actually ingest that commercial BT. And so after that brief introduction, I'd like to just open it up for any specific questions.
Great, thank you, Daniel. Excellent initial start there, and there's already lots of questions coming in. I can see Chris is pretty busy, so I may take a few questions that Chris has asked as well, just so that everyone has a bit of a discussion on them. I think the first one here was that you mentioned BT are used to manage pests with resistance to chemical insecticides. How about resistance to BT proteins? How is that managed?
Yeah, so that's actually a very interesting question. It turns out that individual cry proteins, as I'll kind of describe later, can in some cases bind to entirely different receptors. So for example, resistance to one cry protein may not necessarily confer resistance to another cry protein. And this is really important with respect to genetically modified crops, where those crops will often contain fewer cry proteins than what you would find in the commercial BTs. So for example, in Brazil, the Intacta soybean contains a high concentration of cry 1AC, which effectively controls most pests, but leaves the armyworms out in the field. And so in that case, using a product like an Azawai type strain can actually effectively control those, because as I'll show in a minute, the cry 1C and 1D are so unique from that cry 1AC that they can actually control those insects better. So they can be complementary. But it really does depend on the BT crop, but they can be complementary technologies.
Excellent. Here's another question, a good question here. There are various strains of BT in the market. Will it be more efficient for a region to use their indigenous strain, or doesn't it have a huge difference between strains?
That's also a very good question. The commercial strains that are being mass produced were specifically selected through these screening programs. And I think what maybe Roma could step in on this one, but I think the really important aspect here is the regulation of these commercial products. While it may, in some cases, while there may be other better strains out there in the market, the regulatory rigor, the required, the study that has to go into getting a commercial strain onto the market is immense enough that we really have to pick and choose.
Yep.
Yeah, so Dipel and Zentari, for example, are registered in 65 different countries. So that means, yeah, it's an immense amount of work.
Good answer. And actually, I'll just throw that over to Roma, and then after that, we'll move on because there's lots of questions coming through, but I know that some of those questions will be answered further down, and we'll have time at the end and other questions times. Roma, any feedback on that question?
Sorry, could you just repeat the question?
The question was, there are various strains of BT in the market. Would it be more efficient for a region to use their indigenous strain, or doesn't it have a huge difference between strains?
No, I mean, Daniel's right that so the registration of a new microorganism is going to cost in the region of getting up to, with the product development process and the registration itself, you're looking at a spend that's 5 million plus. So if you're developing a new strain, you have to think it's better than what's already in the market to justify that kind of expenditure.
Yeah.
And then from a biological point of view, there's nothing to indicate that the regional co-location of a strain with a pest, it makes it better. What you can have with some microorganisms is their adaptation to temperature. So for example, if you're working in a really dry country and a hot country, you might want a strain that's better in dry and hot conditions.
Yep.
But a company who's developing the products will know that, and they'll filter for that, and they'll choose the strain, the right strain for the right conditions where they're planning to sell it.
Okay, great answer. Thanks, Roma. Daniel, I'm going to let you go on because I know that we've got lots to get through, and you've got some great stuff coming up.
Excellent. So next, I'd like to talk about the mode of action of commercial BTs. And so we'll start with a video. So commercial BTs work exclusively through ingestion. The caterpillar larvae has to actually consume the crystals and spores. They can't absorb it through their skin. Now these crystals and spores, the crystals will then dissolve in the uniquely high pH of the the high pH of the caterpillar midgut. This releases those cryoproteins, which are actually activated by the insect's own digestive system, which is kind of cool. BTs really use the insect against itself. Once activated, those proteins will then form these pores, which when inserted into the membrane, cause the cells along the midgut to rupture. This allows BT spores and other microbes that are in the stomach to enter the hemocyle, or the insect's bloodstream, and will actually kill the insect through sepsis, through blood poisoning. So in fact, BTs really colonize the insect from the inside out. It's kind of grotesque, but really fascinating. And if you do find intoxicated insects out in the field, you'll find these brown, shriveled up insects because they've essentially been colonized by all of the microbes within the bug. Now, this mode of action is quick to stop feeding, but slow to kill. So this is a lettuce feeding assay. So these are treated lettuce leaves with either Zentari, that's a BTA product, or chlorantranoloprol, the Rynaxypyr technology. This is with second instar diamondback moth. And if you'll notice in the upper right-hand corner, there is the hours or days since application. So I'm going to start this over again. And what I'd like you to focus on is that even after 24 hours, in both treatments for the Zentari and the chlorantranoloprol treatments, insects are still moving around on the leaf surface. However, in contrast to the control, they're not feeding. And that's because when the cryoproteins bind to the stomach and form those pores, it actually neutralizes that alkaline stomach and prevents them from being able to digest their food, to eat anything else. And so for that reason, that feeding cessation will occur very quickly, within minutes to hours after intoxication. while actual mortality, actually killing the insect, can take anywhere from a day or two, because that complete mode of action of colonizing the insect and working its way through their system just takes time, just like it takes us a few days to get really sick after we catch a cold. And that's important to keep in mind because in the case of chemical insecticides, oftentimes you can go out the next day and look to see if you have effectively controlled your target pest. But in the case of commercial BTs and really biocontrol solutions in general, when validating whether an application was effective or not, you really want to give the products sometimes time to work and time to control those insects. Now, BT strains have a very unique protein profiles. This is by far the most important difference between commercial strains. And that's because these cryoproteins are the tip of the spear. This is the primary mode of action of the commercial BTs. Now, you'll notice I have here the two commercial strains sold by Sumitomo Chemical. The one on the left is the dipel strain, the subspecies Kerstaki ABTS 351. And on the right is the subspecies Azawai, ABTS 1857. And if you look, it's kind of color-coded, but on the left-hand side, dipel is characterized by a Cry1AA, 1AB, and 1AC. These are all relatively similar cryoproteins. It also includes the Cry2AA. Now, this nomenclature, it looks a bit strange, but it actually denotes how similar these cryoproteins are to each other. There are in fact hundreds, if not thousands, of different variants of cryoproteins that have been found in nature. And to understand the relationship between these different cryoproteins, we give them these indicator number and letters. So Cry1AA and 1AB are very similar to one another, while Cry1AA and Cry2AA are very different and actually bind to completely different receptor sites in the insect midgut. Similarly, in Zentari, while it does contain the same Cry1AA and 1AB, it uniquely expresses Cry1C and 1D. Now, these actual cryoproteins, that actually defines really the Azawai type strains. That 1C and 1D also have unique binding sites, not only from one another, but also from the Cry1A type protein. So as I mentioned previously about genetically modified crops, insects that may not be controlled by a Cry1AC, for example, can be efficiently controlled by the Cry1C or Cry1D. And that's because different pest species actually have variable susceptibility to each protein. I mean, it's truly fascinating. This table on the left-hand side shows different pest species. And along the top, all of the different cryoproteins that are in commercial BTs really around the world. Most of them only have these 6 toxins. Now, as you'll see, the pluses and minuses represent relative susceptibility. A minus doesn't mean that the insect is not susceptible. It just means that it requires a lot more of the material to kill the insect. And if we focus down on the one on the bottom, the Fall Armyworm, you'll see that while Cry1AB, which is the major component in both the BTK and BTA strains, most BT strains for that matter, can be somewhat effective against this species. Cry1C and 1D are actually acutely toxic to the Fall Armyworm. And this leads to a very fascinating example of where BT subspecies matters quite a bit when trying to control an insect. In general, across other insects like the Helicoverpa or the cabbage looper, the Trichoplusa knee, generally, an Azawai or Kerstaki will control them essentially the same. There's not usually significant differences, but there are those rare instances, like with Plutella and with Spodoptera species, where the subspecies makes an important difference. And just to round it out, the reason for this differential susceptibility is that each insect has actually different receptors. And so that just goes to show that there's really the importance of the lack of cross resistance between some of these is based on that. Now, this is an example of this is a study from Laura Becerra and Associates. in which fall armyworm populations were collected from cornfields across Mexico. So this designation down here along the bottom is actually referencing different cornfields across Mexico. On the left-hand side, you'll see that this is the LC95. So the amount of product that was required to kill 95% of larvae in the study. And so these values can be quite high, but that's only because this is for complete mortality. And you'll notice across the board that while both dipel and Centauri were very effective at killing the insects and controlling these insects, the BTA Centauri was more effective, and that's because of the Cry1C and Cry1D. Now, I wanted to show this also because it really shows how important it is to respect the application rate on the label. Commercial BTs, you'll notice, may have relatively high application rates compared to other chemistries. So you'll be using half a liter of a BT versus maybe only 100 mils of a chemical insecticide. And that's because even though the crystals and spores are very concentrated, you really need to get enough of them out into the field to efficiently cover the whole crop to effectively protect it. And so, especially in the case with commercial BTKs, these products can be very effective against fall armyworm. However, it is important to respect the application rates. While they may seem high, it's only because of this unique property where it does require slightly more of the BTKs to control those insects. So just to be clear, both of these products will work against fall armyworm, BTK and BTA. However, there is this slight difference that does give the benefit to commercial BTAs. And I really only know of maybe one or two other examples where this is actually the case. It's quite fascinating. Now, BTs have an entirely unique mode of action, which is really critical for resistance management. So unlike chemical insecticides that will have typically a single target site mode of action, complex mode of action of BTs means that you have a number of different components that are all working separately. So for example, the Cry1AC, Cry2As will hit different receptor sites, and the spores will also contribute, hitting with a different kind of effect. So commercial BTs are an IRAC group 11 that refers to stomach poisons. And actually, this is a cross section of a caterpillar. So if you cut the insect, across its body, and this is a caterpillar that was treated with a Cry1AC that has a red label to it. So anywhere that you see the red color is actually where that Cry protein bound to the inside of the midgut. So you can see in this case that it's very efficiently covering and will cause all of those cells to swell and rupture. In fact, you can see some of those cells are starting to swell already. Now, this target site is entirely unique from all other insecticides. The only other insecticide that has an effect on the midgut are viruses. And so for that reason, using commercial BTs either in a tank mix or rotation can be incredibly valuable in the long run to protect, really to protect the insecticides that you'll need to rely on even for controlling other insects. Oftentimes, some insecticides will have a broad efficacy across a number of different species. And so even though we may be treating for a different insect, the constant exposure of the armyworm to different insecticidal mode of actions, to a single insecticide mode of action can eventually select for a resistant population. And that's where including a commercial BT in your spray program can help to control those insects because there is no known cross resistance between a commercial BT and any other insecticide class, which is really fascinating and really unique within insecticides. And this is borne out over 50 years of different insecticidal mode of actions being introduced into the market. And so finally, excuse me. And so finally, the commercial BTs work exclusively against the larval stage of the caterpillar. The larval stage of the armyworm. They'll have no effect on eggs or on adults. And that's really full stop. The mode of action is through ingestion. And so it is only the larval stage that can really be intoxicated by the commercial BTs. And so that's why, as we'll discuss at the end of the presentation, timing and coverage are just so important when it comes to commercial BTs. And so we covered a lot of ground there. So if anyone has any questions.
Yes, Daniel, there's lots of questions. There's already 20 answered.
Okay, great.
So
nice
Chris
job.
is really probably getting a sore wrist at the moment, I think, handy, like typing out answers. But a couple questions here. Are BTs temperature dependent, and what is the temperature range to have the maximum efficiency or efficacy?
So interestingly enough, the BTs are not temperature dependent. The caterpillars are temperature dependent. The caterpillars won't eat foliage when it's too hot or too cold, and so for that reason, the optimal temperature is when the insect is feeding.
Okay.
Excuse me. The commercial BTs aren't affected by high or low temperatures necessarily, like from the environment. So it's really has more to do with the insect than it does the products.
Okay, there's another question here around the shelf life. Now you might get, if you're going to cover that later, we can leave it. But there was a question just around what is the shelf life of a BT pesticide, biopesticide?
Yeah, so that'll be very specific to the product. I think it's probably best that you refer to maybe the sales rep or the retail who's selling the products to ask them what the specific shelf life is. Okay. With respect to the products that Valent Biosciences sells, we have a shelf life of at least 2 years.
Okay.
I was fortunate enough to collect some of this dipel DF from our customers in Japan, who oftentimes will think in very long term. This product was over 8.5 years old, stored in a temperature controlled office, and after 8.5 years, still was as potent as the day we sold it.
Okay.
So stored properly, they're incredibly stable.
Okay, stored properly. And so I've got a question here. Yeah, which is always probably the case with lots of things. Daniel and maybe Roma afterwards, but there's a question here from Andy, and thanks for joining us, Andy. It's despite the advantages of BT over chemical synthetic insecticides, one of the concerns is the consistency in their performance under field conditions, which are related to the mode of entry through ingestion and the behavior of the larvae, such as fall armyworm, where big larvae tend to live inside the whorl and are protected. Are there any special efforts to cope or address these problems?
So I think the most important efforts that can be focused on how to get better consistency is twofold. And I'll mention this at the end, but the most important aspects are timing and coverage. A commercial BT will only be effective on the leaf for somewhere between 3 to 7 days, which means that you have to time that application perfectly. And that's where having an organization like this consortium put together by Grow Asia is so critically important to come up with tools that can help us understand the life cycle better and time those applications better. Mm-hmm. I think that's really the #1 source of variability in the field, is that timing and coverage more than anything.
Excellent. Roma, have you got anything to add to that?
No, I don't think I can. Yeah, Dan's right. I think one of the things we should always acknowledge fire control agents is they aren't highly toxic and they aren't highly persistent. So you've got to be really good about when you apply them, really understanding and monitoring your pest population well, so you get the timing of the application of your biocontrol agent with the timing of the pest. So you have to really be good at understanding that biology. Plus, you have to actually have really good application equipment. So really think about what you're doing, about your spray volumes, about you've got good quality tanks and good quality applications. And I think actually this is something that Daniel's probably going to touch on in the second part of his in about a month's time, isn't it? And how to actually get the best out of these products in the field and how to use them well.
Yep, that's right. And we're hoping to bring some video footage as well of actually in the field and how to do that, everyone. So hopefully you'll join us in a month's time. Daniel, if you could continue on, because I know we've still got lots to pack in.
Sure.